Process of muscle contraction. What is a muscle contraction? Muscle contraction is a process in the muscle that leads to either the lengthening, shortening, or even the development of tension in the muscle. Muscle contractions often occur using the sliding filament theory. Nerve transmissions. This is the transfer of a nerve impulse, which is the replaying of a signal that has been coded, to make it travel along nerve cells in order to reach an effector such as a muscle. Neuromuscular junction. This is a chemical synapse formed between a motor neuron and muscle fibre where axons of the muscle nerve meet the muscle. The nerve impulse sent from the brain has to travel over the synapse in order for it to get the muscle to the muscle, causing it to contract and relax. A stimuli such as a rugby ball coming towards you after being kicked starts the nerve impulses because it's a change in the environment which is picked up by sensory neurons such as your eyes. Nerve impulse. In order for it to begin a nerve impulse, also known as the action potential, there needs to be a change in their environment such as the stimuli of a ball coming towards you in rugby. Sensory neurons such as the eyes will then identify this change and send the message to the central nervous system through the peripheral nervous system, which is in charge of transporting sensory and motory neurons from receptors to the central nervous system. The CNS, central nervous system, such as the brain, will then decide what action it would like to make against the stimulus, for example, to catch the rugby ball. This will then start a nerve impulse to spread over a wave of depolarization, which is a rapid change within the cell, along a motor neuron from one end to another, heading towards the muscle fibre. Neuromuscular junction. This is a specific chemical synapse formed between a motor neuron and a muscle fibre where axons of the motor nerve meet the muscle. The nerve impulse sent from the brain has to travel over this synapse. However, the synapse occurs between the synaptic end bulb and the motor end plate. The synaptic end bulb is the end of the axon branching called the axon terminal. Each individual synaptic end bulb may contain many synaptic vesicles which may contain important neurotransmitter substances such as acetylcholine. These substances are vital and responsible to ensure the transmission of the impulse from the axon to the muscle fibres, making sure they occur and are done effectively. The motor end plates are the excitable region located in the plasma membrane of the muscle fibre. It is responsible for the actions occurring across the whole muscle surface. They effectively cause the muscle to contract and relax. The acetylcholine enters the receptor site through the motor end plate and therefore triggers the impulse. When a muscle is at rest before the impulse even reaches the muscle fibre, the ATP, andesine triphosphate, which is a fuel store, is energized and hydrolyzed, resulting in the production of ADP, andesine diphosphate, and a loose P in order for it to bind onto another. The ADP, the hydrolyzed ADP, produces energy in order for the contraction of the muscle to occur. If one ADP is broken down, energy is released, which then can cause the muscle to contract. At this stage, troponin trimyosin is also working as it is covering the actin binding site. This is when the multifunctional proteins have binded onto myosin, allowing it to form cross bridges. To initiate a muscle contraction, an electrical signal travels down a nerve cell, releasing a chemical message into the synapse. The neurotransmitter crosses the gap and binds onto the protein located on a cell membrane. This then causes the electrical impulse in that particular muscle cell. The electrical impulse spreads along the muscle cell and enters cells through the T-tubules. The electrical impulse then opens up the calcium store, sarcoplasmic reticulum, allowing calcium ions to flow directly into the cytoplasm where the actin and myosin filaments are located. The calcium ions then bind onto the troponin trimyosin molecules, which are found in the grooves of the actin filament. Whilst the binding of the calcium ions occur, the troponin changes shape and slides trimyosin out of the grooves, exposing the actin myosin binding sites. This then causes the interaction of the myosin and actin, causing the creation of cross bridges. This then leads to muscles creating a force and shortening. Once the contraction is finished, the electrical impulse is no longer there. Calcium gates close. This causes calcium pumps to be formed, located on the sarcoplasmic reticulum. They remove calcium from the cytoplasm. As calcium gets pumped back into the sarcoplasmic reticulum, calcium ions come off the tropin. This ensures the tropin returns back to its normal shape and allows tropomyosin to cover the actin myosin binding sites on the actin filament. This then means no binding sites are available, therefore no cross bridges can be formed, causing the muscle to relax. When muscles contract, sarcomeres in each myofibril get shorter. This occurs because the myosin and actin filaments slide over each other. For example, when a person is doing a bicep curl, the actin and myosin would slide over each other as the muscle concentrically contracts, pulling the Z line closer together. In order for this to happen, the ATP must be available. The number of times this repeats depends on the number of times you do the exercise. For example, if you were to do 5 bicep curls, it would repeat 5 times, making sure it gets closer in order to contract. However, the actin filament can slide past the myosin filament head, pulling the Z line closer together. In order for a repetition of a muscle contraction to occur, the ATP on the myosin hydrolyzes in order for it to become re-energized. This makes sure the myosin head to repeat the process again with another cross bridge on another actin binding site. To end the contraction of a muscle, the CNS central nervous system stops sending nerve impulses to the muscle fiber. This then causes calcium to return to the sarcoplasmic reticulum, the storage center, and the myosin arm is left energized, meaning it is ready to be used another time. Types of muscle contractions. There are 4 types of muscle contractions. These contractions consist of concentric, eccentric, isometric, and isokinetic. Concentric. During this contraction, the muscles shorten while performing a movement. Ends of the skeletal muscle move closer together, for example when kicking a ball. This happens when the muscle is put against a resistance, like when performing a bicep curl, as the forearm moves towards the upper arm, causing the concentric contraction to occur. Eccentric. During this type of contraction, skeletal muscles lengthen under tension and ends of the muscles move further away from each other, returning the muscle to its normal length. An example of this contraction is where you control the movement when lowering the arm during the down phase of the bicep curl, as this muscle works against gravity to act as a braking mechanism. This contraction is also usually easy to perform as it does not cause muscle soreness as opposed to its sister concentric contraction. Isometric. During this contraction, the muscle length stays the same, or when carrying out physical activity. Tension in the muscle occurs, however, there is no change in the distance between the ends of the muscle. A sporting example of this type of contraction would be when performing the crucifix position on the rings of the gymnastics, or while holding the plank position as the work is put on the abdominal muscles. These types of contractions are easy to undertake, although they rapidly lead to fatigue. Isokinetic. Isokinetic muscle contractions are contractions that happen in a constant speed. A piece of equipment called the isokinetic diameter is used to measure this constant speed and how much force your muscles can generate when contracting.